¿Va? Sí. Hola de nuevo a todos. Seguimos con la siguiente presentación. Ahora tenemos el turno para Marcos Morales, Molinuevo, que nos va a hablar sobre Exploring the somatic landscape of the mitochondrial DNA in renal cell carcinoma using off-target next generation sequencing data. Cuando quieras, Marcos.
Okay, so good afternoon, everybody. I'm Marcos Morales, and I'm going to present my project about exploring the somatic landscape of the mitochondrial DNA in renal cell carcinoma using off-target next-generation sequencing data. Okay. Firstly, let's explain that cancer is the second most common cause of death in the world, but it shouldn't be understood as a single disease, but over 100 ailments as a whole, which share two common characteristics. The first one is an abnormal cell growth, and the second one is the capacity of spreading to other tissues. In fact, metastasis is what kills the most from cancer. So, cancer is normally developed because of a genetic mutation occurred that corrupts the original DNA sequence of a cell. In fact, a mutation is a permanent alteration of that DNA sequence. The genes that acquires that mutation and could lead to the development of cancer are called drivers. Among drivers, we find two main gene categories, which are oncogenes and tumor suppressor genes. Mutations range from single nucleotide variants, in which just one nitrogenous base is replaced, to later chromosomal aberrations or copy variants such as multiploidy. Now we'll focus on the most common kidney cancer type, which is renal cell carcinoma, accounting for around 2% of cancer cases worldwide. Okay. As in most cancer types, it's most common in men and in women, and the chances of suffering from this disease drastically increase after they turn 50. So, depending on their histology, which is the study of the morphological features of a cell, tissue, or organ under the microscope, they can be classified into three main categories. The first one is clear cell renal cell carcinoma, which has its origin in the proximal convoluted tubule of the nephron, as you can see in this schematic image. They often present the loss of the 3p chromosome and sometimes 9p. Among their significantly mutated genes, we find von Hippel-Lindau, mTOR, and TP53. Now, the second most common subtype, englobing around 15% of the cases, is papillary renal cell carcinoma, which has its origin in the distal convoluted tubule of the nephron, and they often present the loss of the Y chromosome in males and multiploidy at chromosomes 7 and 17. In papillary, we find two main subtypes, which are 1 and 2, depending on their nuclear grade. This is the speed its cells grow and divide. Well, their main difference is that types 1 nuclear grade is lower than types 2. Among the significantly mutated genes, we find MER in type 1 and TP53 in type 2. So, third and last most common subtype is chromophobe renal cell carcinoma, which derives from the cell in the collecting duct epithelium, and they often present the losses of chromosomes 1 and 17, among others. Some significantly mutated genes are TP53, P10, and mTOR. Well, as you may have observed, genetic mutations are important to characterize the type of tumor we have. These mutations can be studied through next-generation sequencing technologies, and they will help us to determine the specifically mutated genes of its tumor, and they allow us also to provide a more precise diagnosis and treatment. Okay. Now, talking about genomics, let's recall that there is not only DNA in the nucleus, but also mitochondria. Mitochondria are endosymbiotic organelles, which play an important role in energy production through cell respiration. The mitochondrial DNA is grouped in particular 16,569 base pair long molecules, which code for 37 genes. The number of mitochondria varies depending on so within a cell varies depending the tissue they are in and its energetic requirements. Moreover, it's important to highlight that they present a high mutational rate. In fact, it is 10 times higher than the nucleus DNA one. This is due to the exposure to oxidative damage, their lack of protective structures such as stones, and its strengthless mechanism reparation mechanism. Oh, well. Lastly, I would like to highlight that while 1% of the nuclear genome is codifier, this percentage rises up to 97 when talking about mitochondrial DNA. This could be interpreted as the fact that a minimal mutation in mitochondrial DNA could result, it will certainly result in a protein mistranslation in the future. Well, several studies have shown that some cancer types harbor mitochondrial DNA mutations, such as those in the gene MTND5, which have reported mutations in breast, colon, pancreas, gastric, and renal cancers. However, it is yet to be determined if they act as drivers or not. Well, the aim of my project is to continue the evidence shown by previous studies, which suggested that mitochondrial DNA mutations could make cells prone to cancer development, and study and explore the mitochondrial DNA somatic events of a well-characterized series of renal cell carcinoma tumors by using next-generation sequencing data. I would like to start by explaining the materials and methods. Well, firstly, we extracted both. First, we focus on the tumor sample. We extracted a tumor tissue sample from the kidney by doing a nephrectomy, and we introduced it in paraffin for a better conservation and easier laminating. Blood samples were also obtained in order to determine whether a mutation was somatic or germline. Once the tissues had been extracted, we had to obtain the DNA from them. This DNA extraction and fragmentation was done through a series of enzymatic reactions. Later, some adapters were ligated to those sequences in order to enable us to capture the genes of interest. It's important to bear in mind that our project, the panel genes used in our project, targeted the nuclear DNA genes and not the mitochondrial ones. However, when capturing the wanted DNA, some unwanted debris is also captured. Among the debris, we find the mitochondrial DNA. Well, once the fragments have been captured, they are introduced into a sequencing machine, which will return millions of reads of different letters corresponding to the different nitrogenous bases. Once that, later, the file containing that sequences of letters will be analyzed through bioinformatic tools to obtain the biological information of interest, in this case, the somatic variants. Now, moving on to the results of our study, we find that out of the 142 renal cell carcinoma tumors samples cohort, 118 turned to be clear cell, 13 papillary, and 9 chromophobe. These percentages are in agreement with those shown by previous studies. Moreover, it's also worth highlighting that not all tumors came from primary locations, but 6% of them were obtained from metastatic lesions such as skull or soft tissues. 2% of them became from unknown locations. Okay. So after filtering the data, we just found 2 somatic variants. The first one corresponded to the insertion of a cytosine straight after the adenine in position 10,946. This mutation affected the gene ND4, which codes for NAD dehydrogenase complex 4, which is a part of the electron transport chain. If this mutation had previously been reported in some databases, and if it hadn't been for this, it would have probably been discarded because here we find a null polymeric region of 6 cytosines. These sequences often leads to sequencing errors. Well, the second mutation corresponded to the replacement of a thymine for a cytosine, and it affected the gene CYT, which codes for the cytochrome BCO1 of the electron transport chain complex 3. Well, this mutation had also been reported in hepatocellular carcinomas. Well, and now going through the medical history of the patients, we can say that the sample 208 was obtained from a 72-year-old male who presented a chromophore renal cell carcinoma, and he was treated with a drug called temsirolimus and responded during a 3.3 years follow-up. Well, while the second mutation was obtained from a 58-year-old woman who sadly developed metastases a year after the diagnosis to the soft tissues and bone. Even though she was treated with 2 drugs, on the first line pazopanib and on the second axitinib, she passed away 3 years after diagnosis. Well, we just found 2 somatic mutations. Why did we just found 2 in 142 renal cell carcinoma tumor samples cohort? Well, to determine this, we decided to review the quality of our data. As you can observe in this graph, in the x-axis, they are represented each mitochondrial DNA genomic positions, and in the y1, the average mitochondrial DNA coverage. Coverage is the parameter which counts the number of reads each genomic position has. So as you can observe, the germline samples were poorly sequenced with an average mean value of 5 reads per sample. And well, this low coverage could have resulted in the omission of some mutations because the bioinformatic tools couldn't classify them as trustable. And well, does that mean, so the low coverage mean that we had like a low number of reads? Well, not actually. In fact, as you can see here, the number of reads of the germline samples was even higher than the tumoral ones. However, their coverage was lower. This could be due to the fact that the tumor cells carry more mitochondria than the germline ones, which were obtained from the blood. Now, to decide whether some mutations could have been omitted or not because of the poor germline data we had, we decided to perform a tumor-only analysis on this sample. This tumor-only analysis consisted on discarding the germline data and just focusing on the tumor DNA. Its downside is that we manually had to determine whether the mutation was somatic or germline. To do this, we compared both sequences, and as you can see here, for example, these mutations appear on both the germline DNA and in tumor DNA, so we can conclude that it's germline. Well, after filtering all those data, we just found one somatic mutation. This one corresponded to the replacement of the renal guanine in the DNA sequence for an adenine. And interesting thing about this patient, the patient 141, is that she was a 45-year-old woman, which is too young to develop renal cell carcinoma, and that she also presented a missense mutation in the nuclear DNA in the F8 gene. I said that this is interesting because that gene codes for a protein which is involved in the Krebs cycle of the cell respiration. Therefore, there is room to study if these 2 mutations also were reported in other cases and maybe establish a correlation between them. And well, to conclude, I would like to say that the mitochondrial genome has been widely overlooked in cancer research, but some recent studies are raising attention in this field. even though we used poor data, we found 3 somatic variants. This means that that mitochondrial DNA mutations should be studied further by doing experiments with target mitochondrial DNA and validate the mutations with our gold standard techniques. Because another important fact about our study is that we did not validate the mutations. Therefore, maybe they weren't there and it was all a mapping error. So significant steps toward this would be developing mitochondrial genome databases, designing a gene panel that targets mitochondrial DNA genes. In fact, this is now being done at the CNIO, and the preliminary analysis shows that coverage rises up to 30,000 reads per position on average. And well, lastly, studying the functional role of these mitochondrial DNA mutations in each type of tumor. All in all, we can conclude saying that next-generation sequencing technologies and the use of bioinformatics tools represent an important strategy to decipher the role of mitochondrial DNA genetics in cancer. And well, to end with, I would like to thank all the teachers of the high school for giving me a chance of developing this project, especially Carlos, who was my tutor, and to the Spanish National Cancer Research Center, Javi and Cristina for supervising the project and providing us the data. And well, that's. Thank you all for your attention.
Hola. Bueno, Marcos, muchísimas gracias por la presentación y por tantos datos ahí que nos has presentado. Y bueno, paso a mis compañeros a ver si quieren hacerte algún comentario.
Okay. Congratulations, Marcos. You know, oh, it doesn't work. No. Well, I don't know. Yes. You could do it. You have done that. You got it. I am just going to ask you a general question about your project. You know, I didn't want to go into the heart of it. I just wanted to know, by doing this project, you were expecting to learn a lot of new things, but I'm going to ask you about the things you were not expecting to learn. What have you learned that you didn't expect to?
Well, first of all, I wasn't expected to learn as much as programming as I did, because all this analysis is done through bioinformatics tools, which bases their code on programming languages. And well, maybe also that these mutations can be visualized, and it's not just like I tell you that there's a mutation in there and you have to believe me. In these graphs, you can see where the mutation occurred, which was the mutation. That's it, I think.
Okay. Has this project influenced your future choice of a degree?
Well, maybe yes. I already wanted to say something related to this, but not by informatics specifically. And maybe now it's a door that it's open.
Okay. Thank you.
Well, Marcos, congratulations as well. Relax. So everything it's okay. So first of all, I would like to tell you what I really liked about your job. And it was that you had different kind of results, and some of them were positive results and many of them were not positive. And I really like that you tried to understand and to explain also the negative ones, because sometimes in science, the negative results tell you more things than the good results. So congratulations. And my question is about that. What's your opinion about the importance of sharing data in science?
Well, when internet became available for everybody, the first databases started developing. In fact, now nuclear genome databases stored like quite a great amount of data, but there are just 1 or 2 mitochondrial genomes one. And this made the project a bit more difficult because we had to rely on the difference between here. While if we hadn't had the germline DNA, this project would have been impossible because there was nothing to compare the two sequences. And well, while if you don't have the germline DNA when studying nuclear mutations, nothing happens because you can always rely on the information that there is already shared on databases.
Okay, thank you.
Bueno, pues en realidad nada más. Muchísimas gracias nuevamente, Marcos, enhorabuena y muy bien terminado todo el proyecto. Un saludo.